microbial abundance and diversity. But knowing how many is present, knowing which type is present is not enough. You need to know whether they are active or not. I mean, what kind of processes are they carrying out in the environment? Okay, so you have to measure the microbial activity in order to better understand their ecology in the environment. So as I mentioned, when you measure abundance alone, okay, it doesn't really reflect what's happening because abundance itself is the sum of both growth and loss. So nowadays, as we understand more and more the importance of microbes in the environment, and with the availability of molecular techniques, microbial ecologists are now able to pinpoint specific processes that they want to measure, okay, provided they know the sequence, the gene for the process, okay, the gene that transcribes the protein for the process. For example, nitrogen fixation genes, okay, so if they can probe for the nitrogen fixation genes, then they can later on I'll tell you how they can determine the activity of the nitrogen fixation in the microorganisms. So microbial activity should be measured in order to understand the ecology of microorganisms, because you know that some microbes form spores, they form endospores. Okay. So although uh, an NGS approach utilizing DNA extracted will show the presence of these microbes, but they are not active in the environment. So just because they are there, you see their presence, it doesn't mean they are active and whether they are contributing to the habitat. Approaches to measuring microbial activity as incubation dependent and incubation independent. Okay. So that means for incubation dependent, somehow you have to incubate your samples before you can determine whether any activity is present. But incubation independent is mostly related to the molecular techniques. So for incubation dependent, we can further divide them into cell mass, substrate disappearance, and terminal electron acceptors. Okay, so we'll go through one by one. Okay, when we talk about cell mass, we are looking at the increase of cell mass or cell abundance over time. Okay, so this is to indicate the microbial growth. So incubation dependent essentially means you have to incubate your sample. Okay, so you're incubating a sample, and then you're looking at the cell mass change. Okay, so if you have bacteria in your sample, over time, if the conditions are favorable, more bacteria will grow. So you have a count here and a count here. So if you remember your over the incubation hour. So this will give you essentially your growth rate. Okay. So for example, if you can take the water from Lake, you place it in a bottle, and over the incubation time, you observe for change. So this is incubation dependent observing the cell mass change approach. Okay, is there any problem with this? Do you foresee any problem with this kind of setup? Taking bacteria, you're also taking protozoa, you're also taking other microorganisms inside. So then you know that the protozoa will consume bacteria. So that means this is no longer accurate because there is also growth and grazing here, right? Both processes are occurring. So you will only see a significant increase if the growth is higher than grazing. Okay, so you have to do something to it. What can you do? Okay, if you have grazers inside, you have to remove the grazers. Can you remove the grazers? There are two approaches or two possibilities that you can use to reduce this grazing pressure. One is by antibiotics. You add antibiotics for the eukaryotes. Okay, you add antibiotics for the eukaryotes. For example, your cyclohexamide. Okay, you don't add the antibiotics for the bacteria, prokaryote. So you add antibiotics that are more specific for eukaryotic organisms to either kill the protozoa or to stop their activity. Okay, so that will allow the bacteria to grow normally. Another is separate them. So the term they use is size fractionation. Okay, so you divide your water sample through a filter, usually about maybe 2-micron filter. So the filtrate will have bacteria only. Okay. And then you incubate here. Okay. So that will give you growth with minimum grazing. We cannot say there will not be any grazing because filtration remove most of the grazers, but we won't know whether they remove all the grazers. Okay, but we can reduce the grazing pressure and then we can get a better estimate of the growth. So the first problem is with the presence of grazers. Okay, what is the next? Another problem that that may occur is you incubate it, but in a closed environment. So you might reach a time where the nutrients become limiting. Okay, so you add the bacteria, you size fractionated the bacteria, then you incubated it, so it is growing. But at one time, when the nutrients in the water sample is no longer enough, the bacteria will not reflect the actual growth rate, its actual growth rate. Okay, so what can we do to overcome it? There are a few approaches. First, you shorten the incubation time. Okay. So essentially what you have to do is, first off, when you're doing the experiment, you will have to measure the changes. You have to measure the changes at every hour. Okay. Then when you see the plateau here, this area here, then you know that this is where the growth is no longer linear. The exponential growth of the bacteria, the line that you see here is no longer linear. That means there are some kind of limiting factor that is affecting the bacterial growth. So it is best that you terminate the experiment before you reach the plateau phase. Okay, so you have to maybe shorten the incubation time. Second is you can add nutrients to it. Okay, so adding nutrients to it is, number 3 is dilution. Okay, adding nutrients to it is not so favorable. Okay, in some experiments you can see that scientists will add sugar, okay, to ensure that the bacteria doesn't experience nutrient limitation. But when you add sugar, you are no longer getting its natural growth rate. What you are getting is more of its potential growth rate. Potential because sugar is not naturally in high concentration in the environment. Because it is so rich, it is so nutritious, whenever it is released or whenever it is available in the environment, it is quickly used up. So you don't really see a persistent concentration of sugar in the environment. So when you add sugar to your incubation bottle, you are changing the environment and the bacteria is growing based on a substrate that is not naturally available. So what you're getting is a potential growth rate, not its actual growth rate. So you have to be careful here. What is actually more preferable is dilution culture. Here you mix the one portion of the sample with 4 portion of filtered water. Okay. So your 1 portion of sample has your bacteria minus the protozoa. Here, 4 portions of water where you filter through 0.2 micron. Okay. So the 0.2 micron filter removes all organisms except viruses. Okay, it removes all. So essentially what you're doing is you're adding 1 portion of water with 4 portion of sample where there is no microorganisms, but it has the nutrients here. So you are essentially providing the bacteria here with more available nutrients. But these are the nutrients that are naturally available in the sample. You didn't artificially add sugar, or you didn't artificially add other chemicals to it. Okay, these are nutrients that are readily available in the sample. Okay, so this is called the dilution culture approach. And from there, you'll be able to observe the changes in cell mass, and then you can plot the growth rate of the bacteria. Okay, so we can carry out the incubation dependent observing the increase in cell mass or cell abundance over time to indicate the microbial growth. Okay, of course you have to carry out a size fractionation to remove the protozoa and also to maintain short incubation time. Okay, so that the effects of neutron limitation can be reduced. There are other effects, for example, bottle effects or surface effects that are mostly minor. Okay, what this is concerning is it seems that the size of the bottle that you use in your incubation actually affects the bacterial growth rate. Because a bottle effect or surface effect is based on the principle where nutrients or tend to accumulate where there is a surface. Okay. So a bigger bottle will actually have a smaller surface over volume ratio as opposed to a smaller bottle. So a smaller bottle will have a higher surface area to volume ratio. So a bacteria that is incubated in a smaller bottle tends to have a slightly higher activity than bacteria that is incubated in a larger bottle. Okay, so this is uh, concerning the bottle or surface effect. Okay, but it is when we carry out experiments to determine the surface effect, it is very minimal. Okay, it doesn't really show when you're measuring the cell abundance. Okay, so when you're carrying out experiments like this, you still need about 6 hours to 12 hours incubation before you can get significant difference or significant increase in the cell mass, in the bacterial cell biomass. So for some researchers, this is not, for tropical countries, we need about 6 to 12 hours. So for us, it's still reasonable. But imagine if you are in a temperate country, you're doing experiments during winter, you may need 2 to 3 days of incubation before you can get significant change in the bacterial abundance. So it is not uh, practical to carry out an incubation so long, okay, more than 2 to 3 days. So they have to find other approaches, and what they have decided to do is to use radioisotopes. Okay, to use radioisotopes to measure the bacterial activity. So when we're talking about substrate disappearance, the main aim is the researchers want to shorten the incubation time. So in order to shorten the incubation time, they have to use approaches that are more sensitive. So if you're using cell mass changes, you have to wait until the bacteria grow to a specific and a difference. You need to wait for the bacteria to grow to a certain number before you can actually see the difference from starting to the end point. So you need something more sensitive. So what they used was they used the tritiated thymidine, and also, so these are radioisotopes using tritium. That's why they call it tritiated. Okay, tritiated thymidine, tritiated leucine. So the concept is when the bacteria incorporates this in into their cell mass. So what you do is you incubate bacteria together with the isotopes. Okay. Then maybe after 1 hour or 2 hours, you filter all of it onto a 0.2 micron filter. This will capture all the bacteria here. Okay. Then you wash off the excess tritium. You wash off the excess tritium. The ones that are not incorporated. So you're looking for incorporation. Incorporation means it is being taken into the body of the bacteria. So you remove the excess ones, excess tritium, and then you measure uh, using a scintillation counter the level of radioactivity that is available on the from the bacteria. So the more incorporation, the higher the radioactivity. And because you're looking at radioactivity, so this scintillation counter is very much more sensitive than just counting of the cell mass. So you are able to get significant results even after 1 to 2 hours of incubation. So with this method, they are able to shorten the incubation time and measure bacterial growth using uh, tritiated thymidine or tritiated leucine incorporation approach. Okay, so the question then, when bacteria incorporates thymidine, where do you think it incorporates it in? In what molecule do you think the bacteria will incorporate the thymidine into? The protein. Okay, so you actually, the use of different substrates will actually target different uh, things. So if you use this, you're actually looking at the growth, so as in replication rate. Okay. How fast the bacteria is replicating, is propagating, growing in numbers. However, when you're looking at this, you're more looking at how fast the bacteria is, more of a metabolism rate, more of how it is maintaining itself, maintaining its growth. Okay, maintenance. Okay, so usually now it is recommended that you use both. Okay, you measure both so that you can have an idea whether the bacteria in your sample is growing in numbers or whether it's just maintaining itself. Okay, So that's how you are able, now scientists is able to use radioisotopes to reduce the growth, the incubation time required to measure microbial growth. Tritethyleucine is more related to bacterial survival or maintenance, whereas for thymidine is related to bacterial replication. Of course, initially in the beginning, they used glucose with the 14 carbon. However, as I mentioned earlier, glucose is not reflective of the environment. Adding glucose to a bottle incubation does not really reflect how bacteria will respond, because glucose is too rich and too easily utilized. Okay, so this is the incubation dependent, where you look at the substrate disappearance as an approach to measuring microbial activity. Okay, next is terminal electron receptor. So when you're looking at terminal electron receptor, we are mostly looking at respiration, where oxygen is consumed and CO2 is released. So what we usually do is we can measure either the rate of oxygen decrease or the rate of CO2 increase. Okay. Okay. So one of the most common methods utilizing the changes in dissolved oxygen is the light dark dissolved oxygen method, where the oxygen change can be used to assess primary productivity. This is oxygenic photosynthesis, where the oxygen is being produced. Okay. So the light dark method means one set of bottles are incubated in light, where the oxygen will increase, and one set of bottles are incubated in the dark, where the oxygen will decrease. So we are able to obtain the following results. So when you incubate, for example, your UM-Lec samples, one in a clear bottle like this and one in a dark bottle like this, then you incubate it under the sunlight. So here, photosynthesis and respiration will occur, whereas in the dark bottle, only respiration occurs. So theoretically, the dissolved oxygen in A should increase, whereas the dissolved oxygen in B should decrease. So A shows the net primary production, because this is the net increase in dissolved oxygen after respiration. So there is a net primary production. So if you want to calculate the gross primary production, you just use this slope and you minus the B slope. A minus B will give you gross primary production. So from the light dark dissolved oxygen method, you are able to obtain these results. And if you look at B alone, you are able to obtain the respiration, community respiration of the water sample. I mentioned community respiration because there are also bacteria and other organisms inside the bottle. Okay. The same problems with the earlier incubation experiments that we talked about. To obtain significant results, minimum of 6 to 12 hours is required in tropical countries, but in temperate countries, even more. Okay, so that's why there is also a radioisotope method where they use carbon-14 to measure the incorporation of the carbon-14 into the phytoplankton biomass. So that's how they can reduce the incubation time by using radioisotopes. You have any questions about this culture incubation dependent approaches?
No, doctor.
Okay. So sometimes when you look at the bacteria, so you know that we can do direct counting of the bacteria, and if you use oligoprobes labeled with fluorochromes for specific bacteria, then you are able to determine the different types of bacteria within your sample. Okay. However, this does not represent diversity, partly because you must already know what kind of bacteria you are probing for. So it is not true diversity. You are only looking for specific bacteria within your community. So a modification of this actually allows you to look at the specific genes that are being transcribed, that are active in your sample. So the difference is, instead of targeting the DNA, they target the RNA. So whenever you are looking for activity, if you are looking if you're looking for incubation independent, if you're going for molecular techniques, once you determine that you want to know the activity of the bacteria in your sample, you have to be targeting the RNA molecules. Okay, you cannot do DNA molecules because DNA means the presence of the gene. It doesn't mean whether the gene is being actively transcribed. Okay, so that is an important point to understand. If you're looking for presence, you can look for the DNA. But if you're looking for activity, you have to look for RNA. And with all methods based on RNA, uh, extraction, RNA processing is very tedious. You need very clean methods and approaches because RNA is available, and RNA contamination can easily occur. So once you obtain the RNA, there is always a reverse transcriptase step where you have to amplify or change your RNA into cDNA. That is always a step that you have to do. Okay. So once you have your sample ready, once you have your primers for the specific gene ready, you have to remove the DNA from your sample before you transcribe the RNA into cDNA. And then you look under my epifluorescence microscope to see whether the cells that you're looking at are actively transcribing the gene. Okay. That will tell you uh, whether the gene is being actively transcribed in the population of bacteria that you're studying. However, fish and ISRT fish are very tedious approaches, a lot of techniques to handle. Now most of the molecular techniques used to measure the bacterial activity is dependent upon quantitative PCR or real-time PCR. So just briefly, this is a polymerase chain reaction where you are able to amplify the sequence of interest, the DNA of interest. However, the problem with PCR is you have only an endpoint analysis. So PCR shows uh, presence or absence of the gene, presence or absence of the sequence that you are targeting. Okay. Part of the problem is because of the variable plateau phase. Okay. What you see at the end of your PCR is too variable to reflect the initial starting template. Okay. But they notice that you can get high precision during the exponential phase. So it seems that whenever the cycle threshold here, whenever the PCR product crosses the cycle threshold, the point where the cycle number where it crosses the threshold is proportional to the initial template. So this is what they found out. So at which cycle it crosses the threshold, that correlates to its initial template. Okay, but with these graphs, then you realize that you with a normal PCR, there is no way you can observe for the changes like this. So you need a quantitative PCR or real-time PCR because a Qpcr will have a camera uh, a camera capture system where the real-time changes is being recorded by the camera. So the recording is based on fluorescence. The fluorescence can be provided by using a cybergreen dye. Okay, cybergreen intercalates with double-stranded DNA. So whenever the amplification has occurred, whenever you have more copies, you'll have more cybergreen dye being incorporated. And this one will fluoresce, and you'll have more fluorescence with more and more amplicons. Okay, so that's how you are able to quantify the amplification process. Another step is where you can use a more specific approach, the TechMan probe. This one uses the FRET technology or fluorescence resonance energy transfer. So what happens is you have to have, for Qpcr with a TechMan probe, you have to first generate a suitable probe. A suitable probe is usually one that is annealing to the gene of interest with both a reporter and a quencher at each end. It uses a FRET technology. That means when the reporter and the quencher are close together, okay, the quencher will remove the energy from R, from the reporter, so the R doesn't fluoresce. However, it has a 5' nuclease activity of the Tech polymerase. So what happens is, as the amplification continues and there is a 5' nuclease activity that will degrade the probe. So when you degrade the probe, the R and the Q is separated. The reporter and the quencher is separated. Once the distance between the R and Q is far, the R starts to fluoresce because the quencher does no longer removes the energy from the R. So from that one gene that is amplified will have one reporter fluorescing. So that's why if you want more accuracy, you go for the Techman approach. But Techman approach is more expensive because of the you have to synthesize the probe. Okay, so this is what happens once the R and Q is separated. You have one reporter signal for each new DNA copy. That's why you can have a very uh, more accurate detection of the amplification. This is of course more accurate than cybergreen because cybergreen is not one-to-one, not one fluorescence activity to one amplicon. Okay, whereas for Techman probe, it is one-to-one. Okay, so from here, once you can determine your original template. Okay, so once it crosses the threshold, you can determine the concentration. Okay, of course, you have to calibrate it first. Usually, calibrate it with a known gene from 10 power 0 to 10 power minus 7 to see the where they are, where it crosses the threshold. And threshold is determined as 10 times standard deviation above average baseline, where the average baseline is about 3 to 15 cycles before the exponential phase. So from here, you can determine your threshold and then determine using calibration, determine at each threshold, at each cycle that crosses the threshold, what is the copy number that you're looking at. So from there, you can quantify the number of genes, copy of genes that you have in the original template. So that's how powerful Qpcr now is. This is important, more and more important because most of the determination of infectious diseases, for example, even the COVID-19, they use the RT-PCR approach. Okay, so yeah, so the RT-PCR approach has been used in many environmental studies. Okay, they are able to determine the abundance of specific Shinicococcus species in the sea using Techman probe. Can also study the different types of bacteria in sea, Shinicococcus, Prochlorococcus, and archaea. You can also study the deep sea hydrothermal vents. Okay, so there are more and more applications of Qpcr to study the types of genes being actively transcribed in the microbial community. Okay. So any questions about this? So the use of Qpcr is mostly for incubation independent measurement of microbial activity.